What is mitosis? It is usual to divide it into four main stages for convenience, like four snapshots from a film. Four stages are called prophase, metaphase, anaphase, and telophase. We know most nuclei contain many chromosomes, but here we just show a cell containing only 4 chromosomes for convenience, and different colors are used to show whether the chromosomes are from the female or male parent. In number 1 stages of mitosis, that name is prophase, nuclear envelope disappears. In fact, it breaks up into small vesicles which cannot be seen with a light microscope. And that nuclear envelope reassembles during telophase. So, as a result, diagrams of metaphase and anaphase don't show the nuclear envelope. Okay? Let me show you the difference between the early prophase and the late prophase. In the early prophase, the two centrosomes produced by replication of the original centrosome during S phase of the cell cycle are seen, and in the late prophase, the centrosomes moving to opposite ends of the nucleus where they form the poles of the spindle. In the early prophase, the chromosomes start to appear as the chromatin coils up, becoming shorter and thicker. They are thick enough to become visible when stained, but in the late prophase, the chromosomes are seen to consist of 2 identical chromatids. Each chromatids contain 1 DNA molecule. Also, in early prophase, we can see the intact nuclear envelope, but in the late prophase, we can see the nucleus disappears, forms parts of the several chromosomes. And after prophase, we see metaphase, that chromosomes line up across the equator of the spindle. They are attached by their chromosomes to the spindle. And also we can see spindles that made from microtubules. And each centrosomes reaches a pole. Centromeres helps to organize production of the spindle microtubules. And when the metaphase was ending, the anaphase is starting. In anaphase, chromatids move to opposite poles, and especially centromeres first. And all of the chromatids is pulling by the microtubules. And at the end part of the mitosis, we have telophase. In telophase, nucleolus reforming again, and remains of spindle which is breaking down, and centrosomes will replicate during interphase before the next nuclear division, and a cell surface membrane like other stages of mitosis. And also chromatids have reached the poles of the spindle. They will now uncoil again. Each chromatid contains 1 DNA molecule which will replicate itself during interphase before the next division, and the cytokinesis is happening. This is the division of the cytoplasm and cell into 2 constructions from the edge of the cell. And also the nuclear envelope is reforming like the early prophase at the first of mitosis. Thank you guys for watching. See you soon.